Thank you, and thanks to the organizers for the opportunity to bring some structure to this symposium. So I've been interested in virus maturation in a lot of different systems for quite a while, and the place where I really got my start in virus maturation was bacteriophage. And just a quick, whoa, I'm on a short leash. A quick definition of, or at least my definition, is that this is the process that the virus transitions from a non-infectious and often fragile and spherical initial assembly product to a robust, faceted infectious virus. And the physics and chemistry are well understood for a few systems and reveal some really interesting mechanisms for creating dynamics, shaping energy landscapes, and capturing thermal fluctuations for directional motion through Brownian ratchets. I'm going to give two little vignettes. One is going to be on bacteriophage P22, which is really more of an assembly study because we've been interested in many stages of the process. And the sort of cartoon level presentation of this has only really three major gene products associated with it. In red is the portal protein that makes a dodecamer. And then through some organizational process that we don't understand, we get this procapsid that is guided by the scaffolding protein, GP8, and brings in the capsid protein. Now, this particle is the substrate for the DNA packaging mechanisms, which is made up of two different gene products that pump DNA into this preformed particle with the associated release of the scaffolding protein and the very highly packed double-stranded DNA in this particle. So it's really a liquid crystalline DNA that's packaged in this particle. And then there are associated adapter proteins that are added to the portal, a plug that is inserted to keep the DNA inside, and then these GP9 enzymes that are used for the for getting to the surface and being able to deliver the DNA in the cell. A technique that we use a lot is electron cryomicroscopy, and this is a typical cryomicrograph. And we use image reconstruction methods where we combine thousands of these particles in the same, determining the same orientation. And at the end of the day, we end up with a particle that displays the expected icosahedral symmetry and the organization of the subunits in hexamers and pentamers. And this is a reconstruction of the procapsid. It doesn't have any DNA in it at this time. It's waiting. It has a portal protein. It has a portal in it, but we ignored the portal for the way we did this reconstruction. We can cut this density and look inside. And when we do, we see it's empty. And in fact, in orange, we see some of the scaffolding protein that's still present inside the virus particle. If we do an asymmetric reconstruction, we can find the portal. And this portal is critical because it's where the enzymes attach that burn ATP and pump DNA into the particle. If we take fully mature particles, we can see this kind of fingerprint pattern. Again, we can do a reconstruction where we ignore the asymmetry and just apply icosahedral symmetry. And when we do that, we get a particle that has gone from about 450 angstroms in diameter to 650 angstroms in diameter, but the protein composition is the same. The subunits have just reorganized in a very dynamic manner to create the larger particle that is now filled with DNA, and we can see the DNA very well organized in these layers inside of the capsid. Now, these two images are on the same scale. So this is the procapsid, and that's the capsid. Gives you a sense of the dynamic character of these particles following DNA packaging. We then went forward and did an asymmetric reconstruction where we could see all of the tail machine features of the particle. And here, when we go inside, we can see the portal. We can see individual strands of the DNA as they circle the portal. So the first time I saw this image, I felt like a little bit of a peeping Tom, because it was something that I really hadn't expected to be able to see, but it was a wonderful experience. So this is P22, and the interesting part is, after we did the asymmetric reconstruction, our detailed experimental-based structure looked strikingly similar to the cartoon-based structure. So, and this structure actually came from John King many, many years ago. And when John came and spent some time in the lab and looked at the experimental-based structure, he questioned whether it was worth all of the effort. So, anyway, John is never one to take things too seriously. But somebody took it seriously. We got a nice science cover out of it, and the main people that were involved was, at the time, a graduate student, Gabe Lander, who's now on the faculty at Scripps. Sherwood Kajans provided us with all of the P22 that we used. Peter Privilege gave us a lot of information about the portal and how to interpret our data. Bridget Carragher and Clint Potter used to run our electron microscopy facility at Scripps. They just moved to the New York Structural Biology Center. And Liang Tang came up with the algorithm for doing these asymmetric reconstructions. So that was P22. And now I'd like to just take a few minutes to talk to you about HK97, where we were able to work at a much higher resolution using X-ray crystallography. So HK97 is a lambda-like phage. And Roger Hendricks and Bob Duda at the University of Pittsburgh came up with a very neat expression system where they could express just 2 genes and be able to make these icosahedral capsids that had none of the tail associated with it. That allowed us to grow crystals of the capsids, the particles, and to get a 3.5 angstrom X-ray map of these structures. When we got the structure, we could see every amino acid in the subunits that formed this capsid. This has so-called T=7 symmetry, so there were seven subunits that all had virtually identical tertiary structure. When we looked at the details of this, this was a unique fold at the time we determined this structure, the so-called HK97 fold. And one of the fascinating things about it is that there's cross-links that take place between the asparagine residue and the lysine residue in the structure. The other fascinating thing about it is that structure was determined in 2000, and since then there have been many phage capsid structures and herpes simplex virus, the shell-forming domain. They all have exactly the same fold. So this is the canonical fold of tailed double-stranded DNA bacteriophage. No detectable sequence similarity, but spectacularly equivalent tertiary structures. The cross-links in HK97 create a chain-linked structure that provides an extraordinary amount of stability for these particles, given the high pressure of DNA inside of the particles. And this is a novel topological feature that hasn't actually been seen in any other system except HK97 and its close relatives within the capsid. We've devoted a lot of time over the years to be able to get moderate to high resolution structures of many of the intermediates that exist between the initial assembly product that's formed by the capsid protein and a protease that's packaged inside. The protease processes the capsid protein, takes off the first 103 amino acids to make the prohead 2, which is the substrate for DNA packaging, and then the particle goes on through a number of different intermediates that we've been able to characterize, both by electron microscopy and by X-ray crystallography. The work that was done has involved a variety of people over many years. Roger Hendricks and Bob Duda came up with the expression system that allowed the work to be done. Alistair Stephen and James Conway were wonderful collaborators for electron microscopy. And I think there have been at least 4 or 5 PhD theses that came out of my group of people working on this in one way or another. So I just want to finish with, I don't know whether anybody has shown this, but Roger Hendricks loves to describe the impact of phages on Earth by first discussing comments by Haldane, who says the creator must have been had an inordinate fondness for phage. And then the question is, how many phage are there? And an inordinate affection for beetles. And so then Roger did a calculation that said, well, if phages were beetles, the Earth would be the size of Jupiter. This is assuming 10 to the 31 phages, but this is a couple of other comments. If you connected the bacteriophage on Earth tail to head, they would extend 200 million light years into the universe. And the turnover time for the global tailed phage population is a few days. And the rate of new bacteriophage infections is about a mole per second. So these are very impressive numbers. But I like this, as Roger has done the head-to-tail organization, and we're passing through Eta Carinae. And now here we are going past the Andromeda Nebula, and we've just started on our travel into outer space. So with that, I'll thank you for your attention, and if there's any questions, I'll be happy to try to answer them.